seat, please. So I'm very pleased to introduce our next speaker, Jean-Philippe Lessard. I hope I'm pronouncing that correctly, or JP. Thank you. He comes to us from Concordia University in Montreal, Canada, where he has been an assistant professor since 2014. He held postdoctoral positions at McGill University and the University of Copenhagen prior to that, and he completed his PhD at the University of Tennessee. JP is a community ecologist and also works in the area of biogeography. His broader questions deal with the maintenance of biological diversity, as well as understanding the structure of communities. And he essentially disentangles various processes to answer those questions, including the role of climate, dispersal limitation, and biotic interactions. I was very pleased to learn that he also worked on ants, including kamikaze ants. Those are ants that apparently explode. Is that correct? Okay, so I thought that was a really cool fact. So he primarily uses phylogenetic and trait-based approaches, and that is something he's going to discuss today. He's widely published in various journals, including the American Naturalist, Oikos, Ecology, Proceedings of Royal Society B, and Ecology Letters. And today he's going to be talking to us about improving phylogenetic and trait-based inference of biotic interactions with process-based species pool definitions. Great, so please help me welcome JP.
Thank you for the very nice introduction. So I'm going to repeat what a lot of people have said already, but it's worth saying it many times, I think. This is an amazing opportunity to be here, and I'll be honest, and all the time I was a PhD student at Tennessee, I was dreaming of one day coming here and presenting my work, and here you go. I am, and I'm very proud, and not only proud, but I'm very excited. This is inspiring. It's always a great opportunity to be surrounded by bright people like this that think about similar questions as you do, but in very different, from different perspective. And I suspect that everything I've learned here today will influence my thinking and hopefully my research directions in years to come. So what I'm going to talk to you about today is this thing that Rachel already talked about, which is can we really do anything worthwhile with these phylogenetic and also these trait-based approaches? And as Rachel pointed out earlier, really what we're trying to do with this is to take a pattern and try to infer a process from it. And it's been challenging, and there's been a lot of issues raised with this approach. But then I'll play the devil's advocate a little bit and say that I think there's still some potential in these approaches. And I think that even though we're aware of those limitations, of all the limitations with them, they are being used a lot, right? There's hundreds, if not thousands of papers published using these methods now. And I predict that we'll keep using them because they're easy to use, and they generate patterns that tell us something about the system. We're not sure what it says, but it says something. And so what I've been doing in the last few years is to try to refine a little bit the kind of inference that we can make by looking at these patterns, and try maybe being a bit less naive about what they can do. And also maybe being a bit more careful. All right, so I'm interested in communities and what structures them. And one thing that I'm convinced now of, after many years of studying communities, all kinds of communities, mostly ant communities to be honest though, is that there are multiple processes that are interacting with one another, that are simultaneously acting on communities, and that it is quite a challenge to try and disentangle the signature of these different processes on the structure of a community. And so this is a very simple diagram that I think summarizes a few things that we mostly agree with in terms of what structure communities, which is that species needs to get to the community somehow through dispersal, right? And that they will only persist if the environment there in that community matches with their niche, right? And I'll argue that this is kind of how a species pool is formed, through dispersal and environmental filtering. But then even after that happens, in order for coexistence to happen, there needs to be some mechanisms involved, which often involves biotic interactions. So these things probably all happen. We don't know to which extent they happen. We don't know the strength of these processes, and we also don't know how they vary in space. And I'll argue that really, for a long time, we've been scratching our heads to try to find better ways to infer biotic interaction, and especially the role of competition in ecological communities. And we're still quite far from having convincing evidence that competition is a very important structuring mechanisms in many different communities. But I'm nevertheless interested in quantifying the role of all these different processes in a community. And it's been suggested, and I agree, that this is a very interesting question, that the strength of biotic interactions, or the relative importance of this process relative to others, might vary in space, right? So it might be, for example, more biotic interactions might be more intense or more important in tropical communities and in temperate communities. So it gets complicated. There's many processes. They're probably acting at the same time, but potentially various strength, and the relative strength might vary in space. So how do we handle all this? Because I really think that's kind of the way forward in community ecology, which is to not only try and understand one specific community in one place, but really try to find generalities in terms of which processes are important and how they vary in space. It's not easy. And so when I started my PhD, the work of Cam Webb and colleagues became very popular. And I'll repeat what's been said already today, but it was the idea that we can use phylogenetic information and combine it with information on the composition of communities locally, and then try and say something about the mechanisms that are structuring these communities. And again, it was popular because it was becoming more and more easy to build these phylogenetic trees, and then it was potentially a way to ask questions about the relative importance of competitive interactions versus environmental filtering. So I jumped on the bandwagon and then started using these methods. And really, just to refresh you on how they've been used, is that we can use this phylogeny and information on the composition of our community to calculate a phylogenetic structure. So to ask questions about, are species in my community closely related to one another, or are they actually distantly related to one another? And we usually do this by quantifying this metric called the mean phylogenetic distance. So that's just the pairwise phylogenetic distance between all your species that you then average across pairs. Okay, so that's the unit of comparison that we use when we want to ask if a community is clustered or evenly dispersed. And really, when we do this, we cannot just quantify these metrics. We need to somehow test a hypothesis, asking whether these observed values of MPD are more or less than we would expect given a certain process or given chance, for example. And so what we do is that we will quantify MPD for a given community, and then we want to compare that MPD value to a distribution of null MPD values. And so how do you create a null MPD value? Well, you'll define a species pool that is comprised of species that you think could at least disperse to your local community. Right? And then you'll sample that species pool. You'll sample an equal number of species as what you have in your observed community. And so that'll give you a null community, and you calculate MPD value for that. And you can do that 1,000 times. So each time you put your species back, you pick 5 more, you recalculate MPD. Right? Then you'll get a nice distribution of MPD values. That's what you're seeing here. Right? So on this graph here, you have MPD, mean phylogenetic distance. This is a hypothetical example, obviously. And so your observed value in this case falls between 0.35 and 0.40, okay? And you are interested then in asking how far is that observed value from my distribution of null MPD values. These were created by resampling the species pool, some designated species pool. And so in this case, you find an MPD value that's much higher than all the null created values of MPD. So you can conclude that your MPD value is greater than what you'd expect by chance, so by resampling your species pool. So that's an example of even dispersion, right? Over dispersion of phylogenetic values. And I forgot a slide here, but so what we'll do in general, if we want to compare the phylogenetic structure of many communities along broad scale gradients, because that's what I'm interested in generally. It's not just to ask how is the structure of my community here, it's to ask how does it vary along these broad scale gradient, and does it vary in predictable ways? Then if you want to do that, you need to somehow standardize this measure. Okay, so what you'll do is you'll ask the question, how many standard deviation is this value from the mean of the null values there? And that's a way of standardizing it. And it allows you to compare MPD across many communities. And then when you do that, in fact, what you do is that MPD becomes what you call the net relatedness index, NRI. I forgot to put the equation here. But NRI that tells you not only the value of the MPD, tells you the distance to your null expectation. And that's the metric that we tend to use in this field to compare communities. And positive values of NRI are actually indicating of clustering, whereas negative values of NRI are indicative of even dispersion. It's a little bit confusing, but I'll repeat it as we go along. Okay, so then how do you interpret these values again? So I just mentioned even dispersion or clustering, right? And how it's been done is that you have a species pool here, and then let's say this is my hypothetical community A, and this is my hypothetical community B. And the color, the lines are the species, right? And the colors just indicate the phylogenetic membership in that species pool here. So that's an example of evenly dispersed community, where species come from different clades in the tree, whereas in an example of clustering, the species will come from one or closely related clades. And traditionally, the way we interpret these pattern is that if you find an even dispersion, assuming that niche conservatism is operating, we think that this means that closely related species that are ecologically similar cannot coexist, right? Leading to this pattern, which would indicate that competition is important, right? Whereas if you find a pattern of clustering, again, given niche conservatism, this is the idea that only species sharing a particular niche trait will be able to persist and coexist in a given area. And so one might be tempted to then quantify these patterns in different areas, let's say from the tropics to temperate regions, and try and ask, is it true that competitive interactions, is it true that biotic interactions are more important in the tropics than in temperate region, right? You could do that by trying to plot these patterns for hundreds of communities. And that's exactly what Catherine Graham and colleagues did in 2009. Well, it was not a latitudinal gradient, it was an elevational gradient in the Andes. But it was the first study to really take this phylogenetic structure framework and apply it in a geographical context. And I thought it was really exciting back then, because it was getting to this idea that we somehow need to quantify important processes for communities and to do it along broad scale gradients to try and find generalities. And so that's what she found. She found that at low, well, this is a nice painting by Jan Fietza, by the way, who is an amazing ornithologist and the curator of the bird collection at the Copenhagen Museum. And so this represents a typical clade or a typical community that you'll find in lowland areas of the Andes, and this is a communities of hummers that is more typical of high elevation or highland regions in the Andes. And so what you find is that in lowlands, communities are comprising species from a lot of different distantly related clades, whereas at high elevation, you tend to find more species from one or two closely related clades, which are in fact more derived clades in the phylogeny. And so the conclusion from this paper was, along with the way these patterns were interpreted back then, and still are today, in fact, that competition is the predominant structuring process at low elevation, whereas environmental filtering was the main process involved in structuring communities at high elevation. And so, as much as I was excited about this paper, as much as it got me thinking and in some ways stimulated maybe 5 years of research for me, and asking myself questions about how can we be sure that this pattern that we're seeing in local communities is actually the results of process operating within the communities versus the results of processes operating over the regional pool, which is then trickling down to shaping local communities. All right, so the idea that can we separate the signature of these local processes from the signature of these regional processes on the pattern of local communities. Here's what my concern was about. And so imagine that this is your species pool of hummingbird for all of South America, for example, okay? And I'm interested in comparing the structure of hummingbird communities in highland and lowland, right? And I do find this pattern, which suggests competition at low altitude and environmental filtering at high altitude, right? But then if you take a look at the actual species pool at low altitude versus the species pool at high altitude, you find this here, okay? So if really this is the structure of your regional species pool, just by chance alone, you could get this pattern here. And just by chance alone, you could get this pattern there. So can we then conclude that competition here, for example, is the structuring process, when really all this is reflecting is the structure of the regional species pool, which we don't tend to think is shaped by local interactions, right? And so that was the problem. Regional or local processes, which one are shaping these local communities? And so I sit down with a bunch of smart people, including Jonathan Bellmaker and Jonathan Myers, who's now a prof at WashU, John Chase and Carson Rabeck, and started thinking about ways to try and study communities along broad-scale gradient while being able to separate the signature of processes operating at different scales. And then the idea, well, so that's what you're seeing here. So you got these 4 communities that are represented with black dots, and each of them have a different species pool, right? And just because the community is nested within that pool, you expect certain things about the membership of species in these communities that you could just say because you know which pool they're in, right? So is there any way that we can actually isolate local process or separate them from regional processes? And this is important, and what motivated this paper is that really when you start studying communities in many different places, what you often end up seeing is that the pattern, the processes are very idiosyncratic. So it doesn't look that there is some kind of generalities, but maybe it's because, again, we're just struggle to separate the multiple processes that are operating. So we need to find a way to isolate the different process, quantify the relative importance, and then see if we see commonalities among regions. And we decided, and we suggested this could potentially be done with non-models, but a new generation of non-models that is based on process-based species pool. So what is a process-based species pool? Again, so it's going back to this idea that when you calculate MPD, right, you'll have to compare your MPD value to a species pool to null communities generated from a species pool. But then we need to start thinking about how we define that species pool. Conceptually, first of all, what is a species pool? We should think about that. There's no real guidelines out there for defining a species pool. But also, it has huge implication on your analysis to vary the definition of species pool. That's something that several people have shown. All right, so I would argue that each community should have its own species pool, because there will not be two different communities that will have exactly the same pool of potential community members. It's all context dependent. I mean, of course, communities that are right next to one another will be similar in their pool, but as you go along broad scale gradient, there'll be variation in who can actually disperse there and who could actually persist there. All right? So I say we need to do it separately for each community. And then to improve inference of those MPD pattern, right? And I say MPD, but really everything I'm talking about here could be applied to functional diversity, functional spacing of traits. These exact same analysis can be done with trait or phylogeny. But as a way to infer what we can say about these patterns, how about we're more explicit about how we define the species pool, and we actually model some of the processes we think are important within the definition of the species pool. Okay? I'll show you how it works out a bit later. And then the other issue that we face when defining a species pool is what I call constraint, the level of constraint, which could be seen as a problem of scale, which means, so you have your dot here, this is your community, and you want to know who could potentially colonize this community. Well, maybe it's a radius of 10 kilometers around it. Oh, no, maybe it's all the species in the 100-kilometer radius. No, maybe it's 1,000. We don't know, right? I mean, we can hypothesize, but we don't really know. So what I'm saying here is how about we explore different levels of constraint, different scales along a continuum, and then see how it affects our pattern. OK, so we developed this framework, which we're hoping many people will use, but we'll see how it goes. And the idea is to start with a community-specific species pool, one that's kind of broad, one that we think is probably too broad, but is based on the probability that species can actually colonize this local community. And the way that this is just an example for the community in red here, and we ask what is kind of the broadest pool you could imagine for this community. And we use a tool called dispersion field. It's a macroecological tool that asks, that basically says, if any species, if in this grid cell here, there's at least one species in common with my red community, then let's include that grid cell in the species pool. So you go through each grid cell in South America one by one, and you ask that question. So each of the blue grid cells have at least one species in common with the local or the focal communities of interest. And the idea is if at least one species is there and there, it means that all the others there could potentially get there too. So that's the idea behind the dispersion field. So that's your broadest possible species pool, and it's specific to your community. All right, now what you could do is to create more process-based species pool that rely on different processes of interest. And so how about we say that I'm going to increase the likelihood of including one of those grid cells in South America in my pool for this community if the temperature in that grid cell is very similar to the temperature in that focal community. And that's what you're seeing here. So this is the dark blue grid cells are those grid cells that have a temperature that's similar to the one in the red in the focal grid cell. You can do the same thing for precipitation. You can do the same thing for dispersal, which is in fact here simply geographical distance, so Euclidean distance between each of your grid cell and the red dot, the focal community. And then we also created a fourth species pool, which is the one we call cos distance. And this one is a penalized approach where not only you look at distance between grid cell, but you penalize if you have to go through a big elevational change. So it's another way of modeling dispersal limitation by taking into consideration physical barriers. And so the idea here is that if we compare the result, these MPD results based on these 4 different species pool, we think that if we hit a process that is important, that it will change our NRI value, this deviation between MPD and the null. we think that if we model an important process in our definition of the species pool, it will potentially nullify, or it will strongly affect the perceived structure of the community, so the NRI value. That's the idea. So here we're able to assess how temperature, precipitation, dispersal, and cause distance affect these communities. But then again, it's more complicated because, like I said before, we don't know where to cut that species pool, right? These were broad, okay? So these zero quantile are species pool where the entire dispersion field is included, but then the probability of resampling a grid cell in your non-model algorithm is weighted by differences in either temperature, precipitation, dispersal, or cause distance. So that's a very still broad species pool, but you can further cut them down by only including grid cells that are within your 75th percentile, your 95th percentile, or your 99th percentile. So you go from a very loosely defined species pool to a much more constrained species pool. And so the idea now is that we'll create NRI values for each communities. There were about 250 communities in this case. And then you will do that also for each species pool definition and for each constraint level. And what we expect, again, is that as you increase the level of constraint for these processes, right, you expect to at some point see a reversal in or a big change in your NRI values when you hit the right process and when you hit the right level of constraint. And when you find that strong difference, then potentially you've come across a process that is interesting, and you probably found the scale at which it operates also. All right. So, like I said, what motivated the study was that I was very excited about a paper by Katrin Graham and colleagues published in 2009. And that's the result of their paper back then. Again, you're looking at this NI values where the blue here, so the positive values are communities are clustered, and the red are these values near -2 are communities that are evenly dispersed. There's about 250 communities of hummingbirds in there. And the overall pattern is that as you go from low altitude to the high altitude, you go from even dispersion towards clustering. And we wanted to revisit this data set using this new approach that I've just described to see if these patterns were robust to these new potentially improved approach. And the difference here is that we needed way, way more data. Because when they did this back then, their species pool was simply a tally of all the species that occurred in these communities. Okay? But one issue with this is that it ignores what we call now dark diversity. What is dark diversity? Dark diversity is the diversity that occurs in your region of interest but has not been detected in your local sites. Okay? So there's lots of species that are in this region, but were just not detected in these communities sampled by Graham et al. So what we did in this case is we came back, well, for our approach, we needed to know all the species that were in that region, all the species in each grid cell. So we pulled out a global data set of the distribution of hummingbird. And so we knew basically the distribution of all the species of hummingbird in the world. And that came into our model so that we could use it to define these more realistic and ecologically explicit species pool. Okay, and we also had to get a global phylogeny of hummingbird, which was published in Current Biology by Jimmy Meguiar and colleagues just a year before the publication of our paper. So we had more data on distribution, more data on phylogeny, which got into our model to try and improve the inference from these patterns. And so what we found was this. Okay, so I'm only showing you the results from the temperature species pool because it gets really messy, but the point is I'm showing you only the results from the temperature species pool because applying this temperature filter is the only species pool definition that really had a huge impact on the structure of these communities, right? So we started with a very broadly defined temperature species pool here where most communities, most dots are white because they look random, okay? And as you increase the strength of the filter, you start getting a lot and lots and lots of red dots. And these red dots indicate communities that are evenly dispersed. So that's a pattern of even dispersion that wasn't there at first and became obvious as we increase the strength of the temperature filter. And we did not find this with the precipitation, the dispersal, and the consistent species pool. So this indicated a strong influence of temperature on the structure of these communities. And potentially it is suggesting that only once you account for environmental filtering, you actually can detect this even dispersion pattern, which might or might not indicate the importance of competition. And so this is another way of looking at the same pattern along the elevational gradient. Remember in the original study by Graham et al., at high altitude you had blue dots indicating clustering. Now here you're just finding pretty much lots of red dots a bit everywhere. And so again, it's tempting to conclude that there are two things happening here. There's environmental filtering that seems to shape these communities, in particular temperature filtering. And that once you account for temperature filtering, there's a pretty general pattern of even dispersion that emerges, which is completely opposite to what was found using the same data set 5 or 6 years before. Okay? So this is concerning because there's, like I said at the beginning, there's hundreds, not thousands of studies now that have been published using methods in phylogenetic community structure and trait-based community structure, and that define a species pool very arbitrarily and with incomplete data. And it is concerning that if we revisit these data set with this new suggested approach, which I'm arguing is better, that we'll find a lot of different answers. And so it raises questions about what we've learned in the last 10 years using these techniques. So communities that look clustered with an arbitrarily defined species pool are in fact evenly dispersed if they're accounting for the effect of environmental filters. Right? So we could conclude that both environmental filtering and biotic interactions are important for these communities, and they're acting simultaneously. But to really detect these two processes, we need to use more ecologically and process-based species pool. All right? And then these two processes don't seem to vary so much along the elevational gradient as much as they both seem important all along the elevational gradient. So I think we should care more about species pool definition, obviously. I'm preaching to the choir. But it allows refining interpretation of community-wide patterns of phylogenetic and functional trait structure. Of course, we're still not sure what this even dispersion means. I mean, I completely agree with what Rachel was saying earlier, and there are lots of other things that we need to think about, like niche conservatism. But we can get a bit closer here to a clean pattern, a clean and robust pattern that's not going to change terribly with, let's say, the null model we'll use. Can help detect the influence of multiple processes acting simultaneously to shape community structure. That will be another advantage. And in this case, instead of, it kind of reduces the idiosyncrasy in the patterns and the processes we think are important, in the sense that it did look as if environmental filtering in biotic interactions, or whatever is causing this even dispersion, is acting in many different places. So quickly, future direction. I mean, a criticism that I've been getting from my species pool stuff is that I'm mostly dealing with ecological processes acting now, environmental filtering, dispersal, especially recent dispersal, and biotic interaction. But really, species pool are dynamics, just like communities are. They change through time. And I think exciting venues for research is to try and reconstruct species pool through time. And potentially using the same kind of framework I've presented here today, we could try and pinpoint which change in the regional pool at which point in time switch in the composition of a regional species pool had a huge impact on the pattern we see today. And quickly also, there's Ben Blunder and colleagues that develop a cool framework for looking at quantifying the degree to which dispersal lag might be influencing the patterns we see today. And so this is basically going back to what's the effect of glaciation in the last Pleistocene, and how were species able to recolonize communities at northern latitude. This is something else we should potentially think about when doing these kinds of studies. I'm sorry it's a bit rushed here, but hopefully we can talk about it more later. So with that, big thank to all these people. This was a truly collaborative project with lots of data coming from different sources. It's been exciting. It's been a long ride, and I'm glad it's out now. Thank you.
Thank you, JP. So we have time for maybe 2 more questions before our next speaker.
When you're talking about dark biodiversity, are you talking about diversity that could have been sampled within this community if you had looked at a nearby location? Or are you talking about dark diversity that would have been, for example, other species that are on the other side of the Andes from the community that they were looking at?
No.
Take into account these biogeographic barriers.
No. In this case here, it would be, well, it's a very broad concept that's not that tightly defined. But really what it means in this case here, it's really those species that should be included in your pool that are nearby, but have just not been detected in your sampling effort. Yeah.
I have a question. So your nulls are random draws from a regional pool that match the richness of the local community, right?
Well, they're not totally random in this case because they are weighted by an algorithm. There's an algorithm that goes and resample your species pool with a probability that is proportional to a filter that you apply, whether it's a temperature, precipitation, or dispersal filter.
Right. But when you subset the species that you are going to draw based on temperature or whatever, one thing that happens is that your pool becomes smaller, right?
The pool becomes smaller as you increase the level of constraints, not so much as,
right.
yeah.
So is there any concern about just because you're changing the pool, you might expect the rate of false positives to go up? I actually would intuitively expect the rate of false negatives to go up, so I'm surprised you get so much more power by basically restricting the pool. But have you tried looking at what happens to false positives in a community that has no signal? What happens to detection there just by restricting the pool?
Well, not so much that, but what we've looked, so I mean, first of all, what we're doing here is comparing different species pool definition and increasing constraint on all of them proportionally. So hopefully you're interested in, this increase in constraint, will it have a stronger effect in some process than others, right? So you're comparing on the same basis the different species pool definition. And so in this case, indicates temperature is important because it's the only one that actually does that, right? Now, what we did, though, is to look at whether increasing constraint in the different process had a stronger impact on the size of the pool in some processes. So increasing constraint on temperature, is that reducing species richness of the pool more than with the others? And the answer was no. And as you pointed out, so what you would expect is the rate of false positive to increase with, this is confusing. It's in the supplementary material anyway. but based on the analysis of Nathan Kraft that is done before, looking at how species pool size should affect the power of detecting a false positive or not, it actually suggested that we were not sensitive to that. I just can't remember the direction of the thing. Yeah. Good question. The best question that one could ask, obviously.
Okay, let's thank JP.
Thank you.